more recent, you can see that it looks very different. You have a lot of cases again in Europe, but what is very new are the Americas, both North and South America. And there was a recent publication looking at the last decade of this virus, and you can see the figures here, which are very impressive. More than half of the World Organization of Animal Health member states have reported the disease the last 10 years. 26 had reported the disease for the first time in the last decade. So for still many countries, it has been a recent problem. And they have reported, countries have reported H5 or H7, highly pathogenic AI, and more than 20,000 outbreaks have been reported and 2,000 human cases. So we're really talking about a global disease and a panzootic, actually. So if you look at the virus diversity, so this is a visualization of a global map and some phylogenetic tree, a global one that I extracted from the Next Strain platform, which is publicly available. So if you look at the map in 2009, you can see that there were a lot of clades circulating, so genotypes or clusters, and the clade which is really dominant today is in yellow here. So it was already quite a large clade, but other clades were circulating and had been circulating for already 20 years. If you look at what is in the gray zone here. And if you look at the same kind of visualization, but in 2017, you can see that this clade became highly predominant. There were still some 2.3, 2.1 A and C circulating, especially in Bangladesh and also in a bit in Southeast Asia, and I guess also a bit in India. But apart from this, almost all other viruses which originate from the same ancestor, the Goose Guangdong 96 virus. So this clade 2.3.4.4 became dominant from that year, and then 2 years later, even worse. So something has changed in the virus genetics. But not only in the virus genetics, we also have seen a lot of changes in the virus ecology. So I'm going to present this to you now. First, there has been geographic spread in new areas. I have already showed you the map. Then the situation has been even more devastating in the poultry industry around the world, not only in newly affected regions, but also in regions which were already affected. We have seen a high mortality in white birds. So maybe in new regions, this was not very new. And I know that in India, you have had a lot of mortality of crows, especially the last 20 years. But this has intensified in some regions. We are seeing a lot of cases in mammals, even more than before. And fortunately, only sporadic human cases with this specific clade. So looking first at the geographic expansion, I have already showed this map to you. What is very, very new are the incursions in, especially in South America. And you can see that the virus is at the border of Brazil. And I mean, it's just a matter of time. And we have heard that Brazil now has been affected in a few outbreaks, and on their shores, mostly white birds. And what we see in Europe, where we can expect the disease reporting to be quite transparent, what we see is a huge impact on poultry production with massive mortality and massive depopulation since October 2021 especially. And there has been a huge impact also on trade. A country like France has only been free of the disease one day last year, just one day out of more than 300 days in a year. It's a big issue in terms of animal welfare, with a lot of culling and animal dying from this disease. And the poultry industry in general has suffered a lot in Europe, with a lot of a big impact on economy and employment, and even some hatcheries which had to even stop. They were running out of business, which never happened in the past. And the general public has also been affected by seeing all these this massive depopulation. And if you see here, this is a graph of European outbreaks. You can see that we had a big peak in 2017, and then two others the last 2 years. And what we can also what we see is the amplitude of the peaks, which has really been very high, and also the length of the season of influenza. We used to have several months without H5, but now it's not completely discontinued. But if you look at the last years, really we still had a few outbreaks, not many, less than 100 for Europe, but still the virus has been active. So this graph was extracted from a Chinese publication, where they looked at the information extracted from the World Animal Health Information System, and they divided this circulation into 3 waves. So the first one, which really started when this goose Guangdong virus started spreading internationally, and then a second wave where we mostly had H5N8 circulation. And here the third wave, you can see with this new clade 2.3.4.4, and you can see that the number of birds that have been lost, almost 2 million, and now for sure we are over 200 million, across the world. And this is of course underestimated, because these are only cases which have been reported internationally. Something which is very clear in Europe, I don't know if it's clear also in a country like India, the white birds cases and poultry cases are very much correlated. And so this is a graph from European outbreaks and white bird cases, and you can see that the curves really go together. So what has been very new, especially in Europe, as I mentioned, has been the white birds die-offs. Especially on the shores, we've seen massive mortality of birds, especially started last summer. It was a bit less intensive this summer. The severe situation was in summer 2022. This was really unprecedented. And generally speaking, the number of white bird species that have been found infected has continued to grow, and we are above 32 as of today. What is also new, at least in Europe, is the persistence of the virus in local white bird populations. We are now sure of this. So this is the worst factor for establishment of endemicity of the virus in the region. I also mentioned mortalities and morbidities in mammals. So this is both for sea and land mammals. This is mainly described in Europe and in the Americas. Basically, this is due to wild predators or scavengers feeding on infected birds. And of course, we had sea mammals infected because of this massive mortality of seabirds. And what is also quite concerning is that many of them died from neurological signs, which tells us that human infections could also lead to neurological infections. And most of the viruses that have been found were found with a specific a virus marker, usually in the same protein, in the same gene region. And in some countries, we have seen the report of cat and dogs cases with this publication in China, which is quite recent. And also there was a cat which died in France, which was living next to a farm that was affected by H5N1. And in Poland, they had more than 30 cases of cats which died at the same time. But what was quite strange was their geographic distribution, which was scattered across the country. They have not really identified the source very clearly of infection, but we suspect this is due to cat feed. And what is quite concerning, even more concerning than the other mammals, I mean, these are the mammal-to-mammal infections. So this is how a human pandemic usually starts, with a virus getting adapted to mammals. So we had a large mink farm outbreak in Spain last year, with many mortalities, and they decided to kill all the minks. And the same virus had been detected in wild birds a bit earlier. And they also saw that the virus gained a known gene mutation that again favors mammal-to-mammal transmission. So these minks are very concerning, because they are mixing vessels, species, for different influenza viruses. And you may remember that they were also highly affected by COVID. And so they are having a hard time in Europe, because many of them have been slaughtered, and the production stopped in several countries because of the threat they represent. Now, human cases, fortunately, as I said, this clade doesn't seem to infect human beings very easily. But of course, with influenza viruses, the situation may change at any time with this kind of virus. So we have to be very cautious and try to slow down the circulation in animals as much as we can, in order to protect not only animals but also humans. So WHO still considers the risk as low to the general public and low to medium to poultry workers. And there have been a few cases of this clade 2.3.4.4b in humans, but almost all of them were poultry workers. And some of them had acute respiratory infections, and not all of them died. So why is it a threat? So now this virus has clearly become global, and it has become endemic in many areas where it was not endemic even 3 years before. It is devastating for the global food security, and not only in developing countries, but also in developed countries, and for the livelihoods of people. It has an impact on biodiversity because of the impact on wild birds. And you may have heard that even now birds from the Antarctic are also being affected, and we are seeing some mortalities even there where there are no domestic poultry. And the cases in mammals constitute even a new step to adaptation to humans. And the number of species that this virus is able to infect is just amazing. And of course, the pandemic risk is always there. And what is really constitutes also a big threat is the fact that the situation is not under control in many countries, actually, including in developed countries where vet services had been so far able to control the disease. So in our countries, I mean, I'm talking here about Europe and Northern America, which had been affected already in 2014 and 15, with classical measures which were early detection, early reporting, early intervention. We had been affected several times with white birds introducing the virus in Europe. And so far, Europe and the US had been able to control the disease after a few weeks and with a limited number of outbreaks. But probably because of this virus genetics, it is very contagious. So it is able to move kilometers every day. So it spreads very quickly in bird populations. And it is also infecting now many species. So this virus has a huge ability to move around. And for this reason, so it spreads rapidly. Now it is also endemic in resident white birds, and it infects many species. So there is also high virus pressure all year long. I told you that we don't have marked seasons anymore. It is always active, of course, with big peaks during winter season. And we have seen that it has the ability to enter a highly biosecured unit. In Europe, all genetics have been, all species and breeders have been affected, all kinds. So this virus is really able to infect the whole industry. And classical measures that have been applied so far with really a culling policy, they are not sufficient anymore. So the last 2 years, there have been intensive discussions in Europe, in the US especially, to discuss about vaccination. What should we do? And do we really need to change our paradigm and go for vaccination? So there has been a big change, big movement, especially in people's mind. And there's a general agreement that a new approach is needed to address this new threat. And this new approach is actually mostly based on a vaccination policy. One slide on the Asian situation. So Asia has been facing this Goose Guangdong virus and these outbreaks for 20 years. And so the situation is quite diverse in the region. So you know that China has been infected from the very beginning, and they have the largest diversity of viruses, and any new virus that infects the rest of the world usually emerges first in China. So there's somebody who is quite noisy.
Can he mute his mic?
It's very okay. So China has applied vaccination, as you know, for many years, and they are quite happy with the impact of vaccination. South Korea and Japan are still against vaccination, and like Europe, they've always.
Dr. Abdul Razak, some disturbance is coming from your mic. Dr. Abdul Razak.
Can you mute his mic for the timing?
Kindly put off the mic.
Dr. Reddy, can you mute his mic for the timing?
Okay, I guess we're good now. Thank you. Can you still see my screen?
We can see your slides clearly. We can hear you, but there's a disturbance. Okay. Yeah, now it's okay.
Okay, let me continue then. So South Korea and Japan have not opted for vaccination, but they had massive outbreaks and depletion of millions of birds. So quite a similar situation as to Europe. The Philippines, they have faced this year a very critical situation, and now they have decided to introduce vaccination. In Vietnam, since they've had human cases in the early time of H5 international spread, they had decided to vaccinate birds already for many years. And so the situation is considered as quite under control. Malaysia always opted for eradication. They only vaccinate against H9N2, which they started only recently, like 2 years ago. But they consider that their H5 vaccination is manageable by culling policy. Indonesia has been vaccinating since 2006, and so the situation is quite under control, except that they are now infected with this new clade, which is posing some problems because they already had 2 other existing clades, and they only use inactivated vaccines, which makes the situation a bit difficult in terms of having the right matching vaccines. Bangladesh started vaccinated already many years ago. They consider the situation as well controlled. Pakistan has recently faced many outbreaks, and they are talking about probably introducing the SIVA vaccine. I don't know about other vaccines. And India, I mean, you know the situation better, but there are always some dots on your countries. So we know the virus is around, and you don't have any official vaccination policy. So I'm sorry, this map was updated already, I mean, 6 months ago, but I think it still represents quite well the situation. So you can see in blue the countries that have already allowed vaccination and where actually our main vaccine, Vector-Immune AI, has been registered. So it's an open market. It's just a registration of vaccines and no specific national vaccination strategy. And you can see that some of these newly infected countries have almost immediately decided to authorize vaccination when they saw the immediate impact on their poultry industry. So they wanted basically to save their poultry industry. In countries like the US, which is a huge exporting country in the world, there's still a lot of hesitation, and basically the layer and turkey industries are very heavily affected and would like to vaccinate, but the broiler industry is not much affected, and they don't want any vaccination because they don't want any impact on international trade. So there are a lot of frictions. In Europe, we don't have, only one country started vaccinating, which is France. I will come back to it. But otherwise, Europe has made a big step forward this year by a new regulation, which they updated an existing European regulation. They call it Delegated Act. So every country within the European Union is able to decide whether they want and they are going to vaccinate. And there's a technical annex to this regulation, which imposes a very strict and heavy surveillance program. But otherwise, countries are free to decide on their strategy and on the vaccine technology they want to use. They just can't use live vaccines, but otherwise vaccines of their choice. And I have already presented the Asian situation. So I'm coming back to the vaccine technologies. There are no live attenuated vaccines, of course, but otherwise we have quite a good offer. So there are no immune complex vaccines for influenza, but otherwise the vast majority of vaccines which have been used so far around the world are inactivated vaccines. For some countries, especially China, they use reverse genetic technology, which allows them to make the vaccine low pathogenic before being used, before being produced. And by doing this, they have higher yields of production and less threat to the environment and to people during production. But using this kind of vaccine requires a good matching between the strain, the vaccine, and the field strain. So this is one big drawback. And when we know that the virus evolves every few years, we always run behind somehow. And another disadvantage of this technology is that you cannot differentiate infected from vaccinated animals. And these three technologies really allow to differentiate infected and from infected and vaccinated animals, because they only contain one gene, the hemagglutinin gene or protein, and it means that by ELISA you can, or by HI test, you can differentiate those birds. So in Europe, there have been many trials looking at the different vaccines that exist and trying to compare them, benchmark them. And Europe is very clear on their vaccine requirements, and this is quite new in the world as compared to what countries were using. Their requirements of the different countries. Europe wants vaccines that stop transmission. They don't care about clinical signs, because the birds which are vaccinated and may be infected are going to be killed anyway. They're going to be monitored, and if they get infected, the flock will be killed. So what they want the vaccines to do is to stop transmission within the farm and between the farms. And they want to highly monitor the virus circulation in those vaccinated populations. As a result, they really want this DIVA technology. So these three technologies can offer that, as I said. And for vector vaccines, this is the SIVA vaccine, which has been on the market for more than, around 15 years already in several countries. And then Boehringer Ingelheim has one vaccine, which they also sell in several countries, which is what we call a subunit vaccine. So it is a subunit of the HA gene. It is a protein. And SIVA has also developed an RNA technology, so very similar to our COVID vaccines, but a bit enhanced, because it's a self-amplifying RNA vaccine, which is called Response AI H5. And this vaccine is now authorized on the French market. We have a European Food Safety Authority, and they recently released a very nice report that you can find, if you want, on the internet. It is publicly available. It's a huge report of almost 90 pages, and they make several statements. But the last 20 years, EFSA never even mentioned H5 vaccination anywhere. They had a lot of expert consultations, but never ever on vaccination. And with this new report, they very clearly recommend preventive avian flu vaccination, especially in high-risk transmission areas in Europe. So we don't necessarily need to expand the vaccination to every species, to every geographic area, but we can try to be a bit smarter and try to focus on the high risk. And for example, Italy has a lot of turkey production in the north, so they would recommend Italy to focus on these turkey production holdings. France has a lot of ducks in southwest, so this is also the species which is highly recommended to start with. And the Netherlands have a lot of layers, so they also should start with this kind of production compartment. So the Netherlands conducted a very interesting study last year that they published, but in Dutch. So they studied commercial layers, which had no maternally derived antibodies for H5. These birds were challenged at 8 weeks of age by the, Dutch virus. And they actually looked at 4 different vaccines. There was an inactivated vaccine, which is the only vaccine which has been registered in Europe so far, which doesn't have a good matching with a circulating strain. We also looked at a DNA vaccine, and we also looked at the SIVA vaccine, the herpes virus recombinant vaccine, and also another HVT H5 vaccine, which is a prototype vaccine from Boehringer Ingelheim. And what was interesting was they wanted to study the transmission of the virus. So they infected some birds, and then they put new birds, all the birds being vaccinated except in the control group, but they put new birds in contact with challenged birds, which reproduces better a natural infection, actually, not with a very high dose. And what they found, so these are the infected birds in red. So these are their levels of infection, and in blue you can see the shedding of the contact birds. And so what we can see is that the HVT, the SIVA HVT vaccine gave absolutely no excretion. So basically what they found is that the R0 of the SIVA vaccine, and also the other vaccine, the competitor vaccine from Boehringer Ingelheim, the 2 HVT recombinant vaccines gave an R0 equals 0, so absolutely no transmission of a virus between birds. So this gave the Dutch authorities high confidence in our vaccine, and they have decided to run a large trial based on 700 birds in 3 different farms. And it was started in September. It's going to last for over a year. And it's only in 2 commercial farms. And the birds will be, some birds will be picked up from the farms, brought to the national lab and challenged. And they are going to see if transmission can be reduced. So if this confirms to be effective, then they may go for large-scale vaccination in their country. Now, the situation in France. So France decided to go for only duck vaccination, and we started the 1st of October this year. So now every farm of over 250 ducks has a compulsory vaccination. And the French Food Safety Agency did the same as the Dutch ones. I mean, it was even before what they did in the Netherlands. So we actually had 2. They selected 2 vaccines, one from SIVA and the subunit vaccine from Boehringer. And these two companies were requested to provide the vaccine and help field application. So it was applied on 2 farms for each vaccine. We took care of the vaccination process, and then some ducks were taken to the national AI reference lab, and they were challenged. They were challenged at 7 weeks of age, and another challenge trial was made at 11 weeks of age, because this is the age where the ducks are moved to other places. And they also did a transmission survey. So they challenged some of the birds, and they brought unchallenged birds to the infected ones, and they looked at transmission. And what they found is that for both tested vaccines, the R0 again was below 1. So it means that one bird would not transmit the disease to another bird. So after this, we were able to register our RNA vaccine on the French market, and so was also BI. And there was a big tender, and for the time being, only the BI vaccine is going to be applied, but we're going, hopefully we will be selected in the second tender. And the farmers are also today able to buy our vaccine for a second vaccination, because the government is paying for 85% of the vaccination cost and surveillance cost. And for your information, last year the government invested, it was not really an investment, but spent €1 billion in compensation of the farmers. And this vaccination plan for 1 year is going to cost around €100 million. 1/4 of the 25% of the cost are vaccines, 25% around our vaccination cost on the farms and hatcheries, and then more than almost 40% is due to the heavy surveillance that is imposed by the European regulation. Okay, so these are my take-home messages. First, you have seen that we are really facing an alarming global situation with HPAI, even with HPAI today. And this is an alarming situation for animal health and welfare, for producers' subsistence, for biodiversity, and for human health. This new clade is likely here to stay for a very long time in many countries. Now we have quite a homogeneous genetic virus population, but it's going to diversify again in every country. So it is like a new start from this virus. Several countries with big production are getting closer and closer to vaccination policy. There's a big change in mindset, and the World Organization of Animal Health organized a meeting last year together with another association, and there was a big agreement in the room with people from many countries on the fact that we should go for vaccination, otherwise the world poultry industry is going to die. So for the time being, only France has decided to move on. France is taking a big risk because it's going to lose some international customers for export, but we thought, I'm not a decision maker in this decision, of course, but the vet authorities thought that the situation was too severe and we would lose our poultry industry. So thanks to this vaccination policy, more knowledge and tools are going to be developed on, especially on H5 vaccination effectiveness, and to better know the impact of vaccination on the overall population, and this will benefit everyone. These increases of outbreaks in geographic areas, in animal categories, could be attributed to, and I extracted these different points from a very nice paper here made by very top-level scientists from different countries. So it is due to a continuous increase in poultry production and commercial trade. We know that poultry production worldwide continues to have a high growth. It's also because of increased exposure to wildlife through a repeated annual migration of infected birds. This increase of outbreaks, again, is due to changes in migration patterns because of climate change and land conversion to agriculture production. It is also due to viral genetic evolution, and we saw that this big expansion, it corresponds in time with this emergence of this clade, this new clade. And it is also due to improved awareness, we have to say, and surveillance capacity. So many countries are able to now diagnose very quickly and report transparently. And a potential global scenario that can be very seriously considered is that HPAI vaccination against H5 at least may become a routine vaccination in many countries. And many governments are hesitant to start because they don't know if they are going to stop one day. But maybe this kind of vaccination will become like an infectious bronchitis vaccination or a Newcastle vaccination in the future. With this, I thank you, everyone, for your attention, and I'm open to questions if you have any. Thanks a lot.
Thank you, Gwen, for an excellent, nice presentation, giving an overview of what is the rationale, current global scenario, and the rationale behind the decision to have vaccination in France. Actually, the mandated duck vaccination for HPAI in France actually prompted us to have this workshop, this webinar series on HPAI vaccination, because we want all stakeholders to have a discussion on this particular thing so that we can have a consensus or we can give a scientific inputs to the government of India. That is one of the basic objectives of having this webinar series on this. Is there any questions for her, please? In the chat box, I have 2 questions. What I'm seeing is one is for India. What is the indication? I think it has been shown as not authorized vaccination. I think so. Unauthorized vaccination. It is shown. Am I right, Dr. Gwen? In the vaccination figure in your presentation. I think India and Pakistan, it is shown as unofficial vaccination, right?
Oh, yes, yes.
Yeah.
That's how we consider the status. We know some vaccines are circulating, but there's no official, no official registration of H5 vaccines in India. Is that correct? I mean, is that the status we can give, or?
No, that I think officially we don't have any information on that. Maybe the industry can come up with that. So the second question is about the virus shedding, about the vaccine after the vaccinated birds, the shedding of the viruses in the vaccinated birds. Could you please enlighten us? Because it is stopping the transmission. That means the shedding should be lower.
Yes.
But is the shedding is too low, or the birds are actually, the resistance of the birds has increased, that has stopped the transmission, or whether because the virus excretion itself is too low for the birds to get infected on a mass scale. So that's
it's the same. It's both of them, both factors.
Yeah. Okay. So those are the 2
Nagarajan,
questions.
just a minute.
Yes,
I think vaccine, as we know, even after vaccination, virus shedding will be there.
Yes.
And we cannot say it was low or it was high until unless we carry out some study on the excretion of the virus. Because suppose in India, if you want to do any vaccination for H5, means poultry husbandry practices are very, very wide, very varied. We have few birds to few lakhs birds farm. We have local birds, so many things are there. So perhaps those things we have to take into consideration. And then can you put some point what should be the withdrawal policy once we start vaccination?
The withdrawal policy. So you mean we can't eat the birds for a certain period of time? That's what you mean?
No, about exit strategy.
Okay, exit strategy. Okay.
Yeah.
Oh, this is a very difficult question, actually. First, the scope of vaccination has to be defined. You have to define what you expect from a vaccination strategy at the national level. Do you expect the vaccination to help you eradicate, or do you expect it to just protect some sectors of your production, like in Bangladesh, it's mostly on breeders and layers, so long-lived birds? Or do you expect just to have an overall control of the disease circulation? So once you have established your objectives, then you can think of an exit strategy. And if you consider that the vaccination has done what it was supposed to do and that the situation has stabilized, then you can consider exiting. But personally, I mean, given the global circulation of this kind of virus, I don't see how you could reasonably, I mean, how we can think that if we stop vaccination, we're not going to have any more circulation. You need to be very clear on your epidemiology. Why is your virus circulating? If it is circulating endemically, then it's not due to an incursion of outside viruses. Then your vaccination may help lower your circulation. But you have seen this virus circulates internationally. So I think, personally, I think exit strategies will be a bit difficult to consider. But of course, you need to think of it. Yes. But I think in France, we have started vaccinating, but there has not been any exit strategy defined.
Yeah, no, as you said that there will be trade losses due to present. So same situation, once we start continuing with it, that situation will come.
I mean, I think in the short term, there may be impact on trade, but in the long term, anyway, many countries are so heavily affected that they can't export easily, just because of the fact that they are not free from infection. So I think there's going also to be a change in people's mind.
Dr. Nagaraja, this is Dr. Srivastava from Zoetis. Can I?
Yeah, please go on. Dr. Srivastava?
You have muted me. There's some other disturbance coming.
Yeah, Dr. K.S. Prajapati.
Yeah.
Dr. K.S. Prajapati, could you please mute your phone? Yeah, please go ahead, Dr. Srivastava.
Yeah, Dr. Dauphin, thank you for the nice presentation. What I wanted, seeing the Indian context, the scenario of India, do you recommend we should keep some sentinel birds for monitoring this vaccine? Or we start from a high challenge areas like estuary or a migratory birds area to verify? And one more follow-up question was that how to do a DIVA technology for that. If suppose it is another strain, H5, another strain, like in other cases, we have been using neuraminidase typing and all that to differentiate the dates. If some countries are using inactivated killed H5N2 or something like that. So 2 questions. For Indian context, what do you recommend? We keep some sentinels, or we first start with the high challenge areas near estuary or wild birds density is higher? What is your proposal for that?
Yeah, I would definitely start with high risk areas with birds in contact with wild birds. Yes, I would definitely start from there. And so what do you mean with sentinel birds? You mean having unvaccinated birds on some farms, or having farms which are sentinels?
What I wanted to say how much the protection is given by this vector-based vaccines, how to monitor the efficacy of that. Okay. And the government's pretext is that since it is being a zoonotic importance, they should be knowing this, that whether the infection is running on the farm or not. Okay, so that you can sensitize the workers on that farm.
Yes.
That there is something which you can get also.
Yeah, so usually there's very little acceptance of sentinel birds by the farmers. We know that. And also it's not very, very good to have birds which may suffer from the infection in the middle of a farm. So we're not going to, this is not going to be applied at all in Europe. But it's true that if you have sentinel birds, you don't need to apply a very strict, a I mean, it's a way to easily note circulation of your viruses, because the problem with vaccination is that your clinical signs, I mean, that's not a problem, but that's a fact. The clinical signs are going to decrease, so it's going to be more difficult to see disease circulation. I mean, you can't see it from clinical signs. If you have sentinel birds, of course, you can see some birds dying. But in Europe, at least, it is not really acceptable. But our flocks are going to be monitored every month by an official vet. And so there's such a high, I mean, such an intensive surveillance that we don't need sentinel birds. Now in India, maybe, I mean, in order to save some budget from, I mean, that will be used for surveillance, that could be an option. But again, I think there's a little level of acceptance from farmers.
Yeah.
And then you were asking about our HVT vaccine and how we can check that vaccine is efficient. First, I mean, I didn't take the time to mention that, but what is very nice with this kind of technology is that you can check the vaccine take by ELISA testing. So you run ELISA test against the H5 antigen versus against the NP protein. So this is a non-structural protein which is not present in the vaccine. So if the birds are ELISA NP positive, it means that they have been infected. And another way to check if the birds to have taken the vaccine properly is by PCR, because you can detect the vaccine in the spleen specifically.
Yeah, thank you very much, Dr.
Thank you.
Gwen. Dr. Dey, you have a question?
Yeah,
please.
Good afternoon. My question is, out of the available vaccines against HPAI, do you have any data about whether there is any case of reverse virulence?
No, because none of the vaccines is live. Okay. Yes, I mean, HVT is a live vaccine, actually, sorry.
Yeah.
But it only has the HA gene, so nothing can happen. And it's transformed into a DNA HA. So no, absolutely no cases of reverse virulence, because unless the inactivation is not applied properly on inactivated vaccines, otherwise there's absolutely no risk.
Thank you very much. This is very important to know in the Indian context. Thank you so much.
Dr. Gwen, there is one last question I think we can have. How best the auto vaccine work? One of the question is how best auto vaccine work?
So actually, Malaysia is a nice example of a usage of an autogenous vaccine. They decided to go for an autogenous H9N2 vaccine for the whole country. And so I think they have 2 or 3 providers. Siva is one of them. It's a vaccine which is produced actually in France. We have a very nice virus autogenous production site. So they basically shipped 1 strain to us, and we just provided, I mean, we produce an inactivated vaccine. And so it is actually working very well. They're very happy with it. And the beauty is that they decided on their strain and had a vaccine which was made from their strain. I think this is pertinent for H9N2, which is a virus that does not evolve very quickly. But for H5, this virus moves so much between countries with wild birds. I don't think it is reasonable to rely just on domestic strain. Or otherwise, I mean, it needs to be updated very, very frequently. And new technology vaccines have a broader protection. So a vaccine like this HVT vaccine has so far been tested against all clades, and it has always shown protection. So you have a situation in China where the country is almost using its almost autogenous vaccine base. They have to change the strain every 2 years. but with the new technology vaccines, you have a broader protection, so you don't need to update your vaccine so often. So for H5, I don't think it is a good idea to go for autogenous production, because the volumes of production cannot be really huge as compared to a commercial vaccine. So I wouldn't personally go for that. I would rather go for a new technology vaccine, which provides a very broad protection against several clades.
Thank you, Dr. Gwen. Actually, I have two more.
Excuse me.
I have some questions, but I think we are running out of time. Just one question is that what is the turnaround time you expect for H5 vaccines to be replaced? Number one, or to be updating the vaccine. Number two is what you can. What I want to ask is. Okay, I'll just go with first question because I just forgot to forget the second query. Please go ahead with your first one.
Okay, so this we have two vaccines. So first for the HVT, this one has been on the market for 15 years, so it's already considered as almost an old vaccine. But we've tested it against so many clades, and you've seen the Dutch trials. It's shown a perfect protection. We're trying to see if it would be better with 2.3.4.4b insert, but we don't think it's going to be any better. And the US recently tested it also against recent US strains. It works very well. In Bangladesh, it works against the two clades they have. So there's no current need to update this vaccine. And the reason why it has such a broad protection is that it elicits both humoral and T-cell response. And then the RNA vaccine technology. So this is also the one we had with the COVID vaccine. We are also investing in this technology because with this technology, you can very easily update the vaccine. And in the US, they are even going to use this vaccine for autogenous production, especially for swine flu. And so we don't know yet on what frequency we should update the vaccine. We will see. But this kind of vaccine also elicits cellular immune response. So we expect quite a broad response. So with these new technology vaccines, you don't need to update them very frequently, but it's difficult to give you a time, actually.
Thank you. Thank you so much. Dr. Gwen, it's really an excellent presentation. It is very informative, and hopefully we can have much more interactions with you in future. So thank you so much.
Pleasure.
Now I invite Dr. J.M. Kataria, sir, to give his president's remarks. Kataria, sir. Okay. And I also take this opportunity to welcome Chatterjee, sir, Director, ICAR-DPR, Hyderabad. Welcome, sir. We'll go, sir, Kataria, sir, please go ahead with your opening remarks.
Finishing up Dr. Tosh, I can give it.
Okay, sir. Thank you. Okay. The second presentation in this webinar is will be delivered by Dr. Chakradhar Tosh. He will deliver on the topic HPAI Indian scenario, current status and control strategies. I'll just introduce Dr. Tosh. Dr. Tosh is a eminent scientist who did his graduation from OUAT Bhubaneswar, and he did his master's and PhD in veterinary virology under Dr. Venkatraman. And he's currently working on avian influenza surveillance, molecular epidemiology, host pathogen interaction studies, and development of vaccines since 2005 at ICAR Nishad. Before that, he was working for the development of diagnostics, molecular epidemiology, and selection of vaccine strains for foot and mouth disease in DFMD. His key achievements include detection of HPAI for the first time in poultry as part of a team, and the development and commercialization of inactivated H9N2 vaccines for chickens, and the development of a diagnostic kit for detection of FMD. His research output includes more than 90 scientific publications which are peer-reviewed, and he has participated in workshops and conferences on avian influenza control in more than 11 countries during the past 15 years as part of Y Expert. Sir, I welcome you for this seminar. Please go ahead with your lecture. Dr. Tosh.
Thank you, Dr. Nagarajan, for the introduction, and I'm thankful to Avian Health Professional Association for this opportunity. And I will be talking about the HPAI Indian scenario, and also I will be talking about the viruses circulating in the neighboring countries in the SARC sub-region also. This is the burden of the disease, as you know already in the last previous presentations. It's a threat to the poultry, and we have huge poultry called or killed to control this disease in India since 2000, from the first outbreak since 2006. It is more than 9 million birds has been called or killed to contain this HPAI due to H5N1 or H5N8 in subsequently. And it is also a threat to the wild birds. We have several species of wild bird outbreaks, like crows, a lot of crow outbreaks since 2011. Then we have barred geese, pelicans, serpented stork, cranes, herons, so many species. There are more than 20 individual outbreaks reported from India, and in combination with the domestic poultry, more than 150 outbreaks has been reported and due to the wild birds, and maybe more than 3,000 birds has been killed due to this.
Dr. Tosh, please be close to the mic.
Yeah, and this is also a threat to the livelihood also. If you see that once there is outbreak of HPAI, then restocking of the poultry, it starts maybe after 3 months or 90 days from the sanitary certificate issued by the state government. And there is also joblessness, as you know. This is because of labor-intensive poultry industry, specifically in India, because there is no automation, but very labor-intensive. So there is loss of jobs due to this disease. And this is also has been proven to be a zoonotic importance, because in India has reported one fatal cases of HPAI in one of the children, and it has been reported to WHO. And also it affects international trade of poultry and poultry products also. When there is outbreak reporting from a exporting country, and then the member countries, they feel inhibited to trade with the partner countries when there is a report of HPAI avian influenza. And there is also, this is again economically important disease. There is a lot of government spending on this, including the surveillance and then compensation policy. And one of the data is from the 2006 to 2015. There are more than 20 crores have been spent due to compensation to the poultry owners for the forced culling of these birds to control this disease. And as you know, this is very briefly I'm touching. This is influenza A virus, HPAI, and there are this is a very small virus with the 2 major antigenic determinants, hemagglutinin and neuraminidase. And there are a lot of theoretical combinations of 144 combinations of theoretical combinations of the influenza A virus, and H5 and H7. They are high pathogenic avian influenza. They can be converted to high pathogenic avian influenza. And this is the birds, are the wild birds, or the aquatic habitats, are the primordial host of influenza. All the influenza A viruses, when it jumps to poultry, domestic poultry like chickens or turkeys, they get adopted. And specifically, if you see the low pathogenic avian influenza like H9N2, they are very self-limiting diseases, and the symptoms include maybe there is loss of production in the farm level, but in laboratory condition, this does not kill the poultry host. But in high pathogenic avian influenza, where there is fatal disease, and it may go up to 90 to 100% fatality. And these are the notifiable diseases, H5 or H7, any N combination are notifiable because they can be converted into high pathogenic avian influenza, and also they are zoonotic importance. if you see the outbreaks since 2006, we are reporting almost each year outbreaks due to poultry in combination with other birds like crows or other birds. But the last 1 year, if you see the outbreaks, the first outbreak in October 2022 started in Kerala, Alappuzha district on 16th October 2022. And there is huge mortality of ducks. These are some Chera Chambali ducks breeds here in the paddy fields after the cultivation. And there is corneal opacity. You can see the blue eyes, and there is torticollis. And the samples are sent to Nisad Bhopal for testing and confirmation. And this turned out to be influenza A virus with H5N1 subtype. And subsequently, this virus was detected in other districts like Kottayam and Thiruvananthapuram in poultry farms or backyard farms in dogs. And more than 20 epicenters has been declared in Kerala itself. And then subsequently, we have the Jharkhand in Bokaro. There is a poultry farm where there is H5N1 is detected. And in Bihar, two districts H5N1 detected in 2023 March. And very recently in September, we received samples. The mortality started in September, and we received the samples, tissue samples in October, first week of October. And these are two dead birds. One is black swan and another is silver pigeon. They are thought to be positive for HPAI H5N1. And all these viruses, we went for sequencing and the analysis of these viruses and try to compare these viruses with the neighboring countries or the global database. And all the viruses of H5N1, they were highly pathogenic to poultry based on the HH cleavage region. And we did a phylogenetic analysis of these outbreaks. And the viruses, the dog viruses, they grouped with the clade 2344B, what is currently circulating across the globe, including, and these viruses are related to the contemporary isolates in the phylogenetic tree with the viruses circulating in Eurasia, that is Europe and Asia, African region, and also North America, same 2344B. And however, the chicken isolates in Jharkhand and Bihar, they belongs to 2321A, which is circulating in the contemporary isolates of Bangladesh and India. So that indicates that the 2344B could be a new introduction to this country through wild birds in migratory birds, and that coincides with the bird migration, because we know that there are a lot of millions of birds migrate from Arctic region to Indian Ocean during October and reach by September and October, and they go back February or March. So the outbreaks coincides in Kerala in October. So that indicates, based on genetic analysis and the time of the outbreaks, that indicates the wild bird migration could be one of the causes of the spread of this virus. And the chicken viruses, they are related to these viruses circulating in Bangladesh and India, and they could be, the virus could be persisting in the environmental environment that indicates the persistence of the virus in South Asia. This 2321A, which was isolated for the first time in 2010 and 11. This is the overall picture of the viruses circulating in India since 2006. We have since 2006 past outbreaks. There are almost 382 outbreaks reported from 24 states and union territories. These are the genetic clades emerged. The past outbreaks in 2006 was a Qinghai-like virus, that is clade 2.2. And that persisted till 2010, because subsequently we didn't find that, detect that virus from the samples submitted for confirmation, or the active surveillance samples. We didn't find that clade 2.2 virus in the samples. However, in 2011, India reported 2321A clade And this virus was, the earliest virus was 2010 virus in wild birds detected in Bangladesh. And that virus is still circulating in this SARC sub-region. Then we detected a new clade 2321C virus in 2014 and 15 in 3 states in Kerala, Chandigarh, and Uttar Pradesh. And that virus, we are not getting any detection of that virus after that period. Then we got a new subtype of H5N8 in 2016 in 7 states and Union Territories, 2344B. That, of course, it is H5N8 virus. And the H5N1 virus 2344B was detected in 2020. And this says this poultry outbreaks. India has reported a human infection, a fatal human infection due to clade 2321A. And this is if you total overall scenario, if you see the outbreaks from 2006 to 2023. This is the data reported from India, and we have compiled, and most of the activities are from October to April. And the activities peaks in January, the high-path influenza activities. Peak is reported in January and starts in October. But however, there are some deviations, like there are reports from April, May, June. One outbreak was reported in 2007, in July also from Manipur. So this is the overall scenario of the Indian outbreaks, high-path influenza outbreaks in poultry and wild birds. That also concerns the poultry, the red one, and the wild birds, the blue ones. When there is a wild bird outbreaks, there is also poultry outbreaks has been reported. We have seen the antigenic divergence because we have seen a lot of clades changing, and we wanted to see how these clades antigenic divergent or not as a preparedness for future vaccine. So we have 4 clades 2.2, and there is a 4th order clade of 2.2, that is 2.2.21, and we have 2321A and 2321C. We have, it is before the study was before the, and we are studying it with 2344B. The data is not yet out. And with all the chicken antiserum against 7 isolates from all the 4 clades, and we see the antigenic divergence between clade 2321A and 2321C. And these 2 clades are again more divergent from the earliest clades, that is the 2.2 clade, which was found circulating 2006 to 2010. And so this is the antigenic diversity that indicates one clade vaccine against one clade may not give optimum protection to the birds to the divergent clades. This is the some scenario of the SARC sub-region, and the outbreak started in India in 2006, that is in the SARC, and then we have the outbreaks in Pakistan, then Afghanistan in poultry and crow, then Bangladesh in 2007, Nepal in 2009, and Bhutan also reported. Out of 8 member countries, 6 countries have reported the HPAI outbreaks in different time periods from 2006 to 2010. However, the island nations like Maldives and Sri Lanka has never reported any HPAI in poultry or any wild birds. So total outbreaks, if you see, there are more than 1,400 HPAI outbreaks from these 6 countries in this SARC sub-region. H5N8 reported in India and Nepal. Of course, there is outbreaks from Pakistan also from H5N8, and there is human cases from the neighboring countries, from Bangladesh, Nepal, Pakistan, and India. So this is overall of HPAI situation, and the virus is still circulating in the neighboring countries, and still it is a problem with H5N1 and also. And H5N8 we have not reported since 2017. This is a phylogeny of the SARC sub-region. You see the 2.2 virus, the lowest panel was found in 2006 to 2011 from all these neighboring countries, and after 2011 there is no report of 2.2 viruses. from SARC sub-region. Then there is a 234 virus that was reported in Bangladesh 2011 and 234 for H subclade that is reported in 2020 only. Then the recent 2321C was reported in Bangladesh, India, and Nepal, and that is within the period 2010 to 2015, and that virus is not reported after that. Then the first report of 2321A was in Bangladesh, that is in a wild bird, migratory bird, but in poultry outbreaks started from 2011, and it is still continuing in Bangladesh and India, but Bhutan and Nepal, they have reported. After 2013, there is no report of 2321A. Coming to this new clade 2344B, which was reported for the first time in India 2016-17, due to H5N8 virus. Now it is H5N1. The N8 has been replaced by N1 gene of N1 gene, and it is still circulating in India and Bangladesh. So this is the overall status of HPAI in SARC sub-region. The virus is still persisting, and new introductions, because the virus already presented by my previous colleague, Dr. Gwenaëlle, that the virus has gone to the wild birds. So the birds are migrating, so it is very difficult to eradicate the disease, and so so we have to be prepared for this virus. What are the risk factors for this spread of this virus in India specifically? I'm talking India and the SARC countries. It's the wild bird movement in Central, because India, the SARC country, comes under the Central Asian flyways between, and the viruses they migrate from Arctic region till Indian Ocean. During September, October, they reach, and then they stay here, and they go back by February and March. We have a lot of wetlands, because this virus, these birds, they come and stay in the wetlands. They are called also sometimes. There are 75 Ramsar sites in 23 states and union territories. So they stay here for this period, and then go back by February and by March end. And then huge poultry population. We have 851 million poultry with at least 40% backyard with the, as per the livestock census, 20th livestock census. And there is a low or there is limited or no biosecurity in these farms, and the biosecurity varies from farm to farm. And there is a huge live bird markets, and there is market chains, and there is very few or low biosecurity practices in these live bird markets, and the market chains supplying the foods. Then we have huge dog population also, and as you know that these dogs are sometimes play a role in the carrier of the virus, and it has seen they shed virus up to 17 days, also sometimes without showing. And these clinical signs in dogs are limited compared to the chickens. So they are also play a major role in the sustenance or the persistence of the virus in this SARC sub-region, and also in maybe Indian subcontinent. Then we have huge international land border, more than 15,000 kilometers of land borders, and some land borders with countries, neighboring countries, they are porous. So there could be informal trade between the countries, which is helping in the spread of this virus or the persistence of the virus in this sub-region. And then we have seen a lot of publications also. Bird migration is playing a major role. And the earliest report, like 2008 to 2010, by publications indicate there are 3 times the virus has been introduced to West Bengal. The outbreaks in 2008 to 2010. And wild birds were suspected to have played a role in the spread of spread of this virus in West Bengal. Then one of our publications said when we got a new virus 2321C in 2014 and 15, we tried to compare these viruses, and based on the medium joining network, we find that the outbreaks one of the outbreaks in Kerala is linked to the wild bird virus from the Middle East, and the other outbreaks were linked to this initial outbreaks. So wild birds could be the probable vector for the transmission of this virus to the poultry. The H5N8 virus we see first time in 2016 and 17 in 17 states and the Union territories, in zoos and in wetlands. So started in October, and based on the genetic analysis and the time of the outbreaks in the Indians of India, that indicates that the wild birds could be the probable source of the virus. And this virus are very closely related to the viruses found in one of the lakes in either in Qinghai Lake in China, and another lakes found viruses found in wild birds in the Mongolia and Russian Federation, that is Utsuro Lake. So that indicates the wild birds or migratory birds play a role in the transmission of the virus, spread of this virus. Then informal trade, as earlier I indicated, could be the one of the reasons for spread or the persistence of the virus in this sub-region. The control strategy followed because we have following the outbreaks, initial outbreaks in 2005, we have the Government of India formulated an action plan for national action plan for prevention and control of avian influenza, and based on the experience gained from the outbreaks, this action plan has been revised from time to time, and the latest one is 2021 revised. And India uses test and slaughter policy, no vaccination policy, no vaccination allowed. And the action plan has 7 chapters, including different actions to be taken, and then actions during outbreaks, what are the preparedness, and then post-operative surveillance, declaration of the country freedom. And then we have also guidelines for zoological parks following the outbreaks in 2016. We devised these guidelines for zoological parks, and now the 7th chapter is compartmentalizing, because we see there is every year there is outbreaks. So that is one of the strategies has given for even in the presence of outbreaks, the country can trade with the partners for poultry and poultry products. So for the surveillance and the control, we have, these are the laboratory networks, 5 regional disease diagnostic laboratory at the different corners of the country. Then we have CDDL, then national and OR reference laboratory at Bhopal, Central India, at ICAR-DPR. We have a human influenza laboratory at National Institute of Virology, Pune, Maharashtra, and we have state diagnostic labs, more than 256 state diagnostic laboratories for this activity. And this is how the sample flow in case of active or passive surveillance. The disease investigation officer at the district level is the nodal officer for collecting the samples and then sending and intimating the state director animal husbandry. The samples are referred based on the emergency, referred either to the R details under their jurisdiction, or if it is their emergency, they sometimes directly refer the samples. If it is close to the R details, if it is tested positive, then for confirmation, they send the samples with the reports to the NISAD at the Central Reference Laboratory, National Reference Laboratory for confirmation. And then it is, after the confirmation, the results are communicated to the Department of Animal Husbandry, Government of India. And then following this, the reports for HPAI, then there is an action is followed, setting up control room in the state where this outbreak has been reported under the control of the district administrator, and then constitution of a rapid response team, and then demarcation of infected zones and surveillance zones. Infected zones 1 kilometer radius of the epicenter, and surveillance zone from 1 to 10 kilometers. And then restriction of the movement and ban of sale of poultry during this the infected zones. Then there is a campaign of education and communication regarding this HPAI. Then within this infected zone, depopulation of the birds and payment of compensation for forced culling of the birds within 1 kilometer radius. Then after culling operation, cleanup, disinfection, and then sanitation. And then the sanitation certificate is issued by the state government, and then post-operative surveillance plan is initiated from 1 to 10 kilometer radius, and 4 fortnights, every 15 days interval, samples are collected from all these 10 kilometer radius. And this entire process of next restocking takes around 3 to 4 months. And there is no vaccination policy in India. And this is the sample submission address for influenza viruses. And India has gone for compartmentalization of against avian influenza for continuing of trade in the presence of presence of high-path avian influenza in the country, in poultry. And there are recent reports that WA has approved 26 poultry compartments in 4 states for continuing of trade in the presence of outbreaks in wild birds. And when there is another chapter that is when there is a suspicion of high-path AI in zoo or wetlands. This is the chapter 6. There is no calling of birds is necessary because all the details are given in this. And unlike poultry, it is not advised to call the birds. Of course, the sick and other birds, they can be called. And the samples are sent after these birds are segregated. The sick birds and the healthy birds are segregated, and a biosecurity and biosafety measures followed to control this, to separate this infected and the healthy birds in zoo or wetlands. And unlike POSP samples in poultry, same way the samples from the wild birds fecal materials or if possible cloacal swabs are submitted for confirmation for the testing of the disease. If 2 subsequent consequent attempts found to be negative in the samples, then the zoo is open for the public. The outbreak is declared closed. And because we have seen the outbreaks in poultry and wild birds, we've seen the outbreaks in the infection in people, and also environments like wetlands and live bird markets also have been reported with high-path AI. So this is one of the best examples to address the issue under One Health strategy, because as per the One Health, it is the health of human, animal, and environment interrelated. So we cannot ignore anyone. So this is the best example to study under One Health approach. And what are the opportunities India has? These are because we have a proposal for a National Institute of One Health in Nagpur, and we have interministerial task force and joint monitoring group established at the national and between the ministers of animal health and environment and animal husbandry. Then we have National Standing Committee on Zoonosis and One Health Consortium launched by DBT. Then we have the veterinary services have been evaluated by performance of the veterinary services by WA, and already the report has been submitted, and we know the strength and weaknesses, and the recommendations has been given in this report, so we can improve upon for proper control of this influenza. Then we have the political backings for this One Health approach in the G20 declaration in September 2021. And then we have the surveillance softwares like in human IDSP, NADRS, and NADRS surveillance systems. Only thing we need to be linked all these systems so that we work in a coherent manner. And we have already a joint rapid response team when there is outbreaks reported in animal health or poultry. Then it is intimated to the public health, and there is joint response teams or the surveillance of outbreaks areas by this animal health and public health team. And this is as a preparedness. We have the NISAID has developed reverse genetics vaccines against H5N1. Of course, this is the earliest cleared circulating in India, that is 2.2, and also it has been extended to 2321A. And the protective titer, the single vaccine, the protective titer more than HIV 128 remains till 20 days, whatever the study period. So as a preparedness, we have NISAID has developed this vaccine for chickens. To conclude my talk, this is that HPAI is difficult to eradicate in near future because the virus has gone to the wild birds and the water birds. So it is very, very difficult with the current scientific knowledge. It is very difficult. So it will stay for some time. And then there is need to control this disease. There is need for One Health surveillance proactive rather than reactive approach to assess their impact. Then we need to enhance the biosecurity measures so that the spread or the contact, the spread of the virus is limited. And the vaccine has been used an additional layer of protection. It is not exclusively only vaccination will give a complete protection. And besides this vaccination, the other measures like biosecurity, surveillance, and then monitoring following vaccination, because vaccination starts, the stakeholders should think that vaccination is not the end. The vaccination is the starting point. Till the sale of the product like egg or chicken or meat, it is the responsibility of the stakeholder following vaccination to monitor, because we know there are other issues, technical issues with the vaccine. The spread of the virus, of course, the vaccine suppresses the clinical signs of death, but still the virus persists in the environment. So it is the stakeholders who has to own the responsibility following vaccination to monitor the virus. And then because without monitoring the vaccine, after some times the vaccine failure has been seen in other countries like China, but China very promptly, they have replaced the vaccine strains. And they have successfully controlled the disease. And they have brought the disease under control, and also the human outbreaks of H7N9 with the trivalent vaccines. So the vaccination is not a magic bullet. It is the starting point, as I told, that the responsibility is with the stakeholders following vaccination. They have to monitor the virus spread till the end of the sale of the products like eggs and meat. So thank you very much for your patient hearing. Thank you.
Thank you, Dr. Tosh, for an excellent and informative presentation. Now, there is one question from Dr. Milind Limai. What would be the government policy to control if no vaccination policy is in place?
No, vaccine, as I told, vaccination policy, vaccination is a complex issue. Everybody agrees, you agree, and I agree, because there are technical issues, there are logistical issues, and there are policy issues. And if you see, there is a 2006 notification from department that the state or the stakeholders, they should approach to the Department of Animal Husbandry for vaccination and what for they want and how many doses. So it is the stakeholders or the states, they have to approach the government of India for this for vaccination. Then as I told that there is issues like technical issues of which strain to include in the vaccine, and then the monitoring of the vaccine, the following vaccination, and then the vaccine matching protocols. So there are so many technical issues. Then there are policy issues, exit strategy, and as Gwenaëlle told that it is very difficult to develop an exit strategy. But there are some policies need to be developed. How long we want to go for vaccination? It is like IBD or ND. Then there are logistical issues also, and that has to be addressed. And it is, I think, it is the stakeholders of the state who are going to vaccinate. They have to approach the department for a development of policy, for a suitable development of policy, because so far there is no vaccination, but globally people are going for vaccination. And as per the Animal Health Forum this year in OA, there are specific meetings for this avian influenza for 2 days, and they have advised the governments to work in coordination with the stakeholders for the development of control strategy. And I think there is a need for the stakeholders to approach the department for the development of policies for subsequent policies for control strategy, besides the comprehensive strategies followed currently.
Dr. Nagaraja, can I give my points here?
Yeah, please go
Yes,
ahead.
sir. As I think the very objective of this seminar is, see, we don't want to influence the government policy. We'd like to work with the government to have a constructive policy. But as a scientific community, we feel that vaccination against HPAI is one of the essential tools. However, such a magnitude of disease cannot be controlled by just vaccination. So a combined efforts of compartmentalization, biosecurity, vaccination, reporting is a kind of a part of the approach. And of course, as a forum under IAVR, we like to work efficiently on the, what is the, like I have seen the question, that what is the extent of damage vis-a-vis improvement post introduction HPAI vaccine in Bangladesh. I do work very closely with that country for my business purpose, and government has also a lot of data. So we like to substantiate, facilitate, and equip the policymakers. Then it's up to them to take a decision. But whatever we do, we like to work scientifically for the best interest of India. And if a vaccine can play a role, but it cannot be the ultimate tool. It is one of the tools. That's the point I'm trying to make. Thank you.
Shall I ask one question, please? Dr. Mujibathar from Hyderabad.
Yes, sir.
Please go ahead, Doctor.
I have got two simple questions, very practical questions. Number one, I am interested to know how many wild bird samples have come positive and which species, which year? Hello?
Yes, in my past slide, if you see, because here I have not shown the very detail of the, you see, these are some of the wild bird species like crows. Then it is 2008 also we have seen crow outbreak samples from Assam. Then from 2011 onwards, we have seen crow outbreaks in 6 to 7 states. Then we have bar-headed geese outbreaks in Punjab, then pelicans and painted stuff. Then crane outbreaks from Rajasthan, and then swans and these pigeons from Maharashtra. But I don't remember now. I cannot give. I don't have data here. I have not kept this data. But we have received a lot of wild bird samples for testing and confirmation of the HPAI, H5N1 and H5N8 viruses.
My second question is, sorry for to have some different type of a question. Since 2006 to now, already almost 2 decades are over, more than a decade, we are sticking to no vaccination policy. What is this? Listen to me. When in the year 2006, first outbreak occurred in our country in Nawapur, I was also part of that discussion. At that time, one secretary, Madam Upama Chaudhary, was there. Vaccine was lifted from New Delhi to Nawapur, and after discussing with so many people, it has been withdrawn. We have not gone for vaccination with so many reasons, because restocking will take longer time and so many disadvantages rather than advantages if India goes for vaccination immediately. That's how we skip the vaccination at that time. But now, sorry for asking this very bold question, keep your hand at your heart. and tell me it's not only one vaccination, irrespective of the state, 100% poultry farmers are doing vaccination 5 to 6 times, and it's giving very good protection. I don't want to elaborate. I don't want to comment on this issue. I missed the topic of that France lady because of some connectivity. She was talking on behalf of Siva vaccine. Wonderful discussion. When we are talking about no vaccine policy, how can we import good vaccine to our country? And we are just leaving the farmers to some XYZ type of things. I think it's not fair. It's high time that we should have a real strategic plan. Every year, hell of a lot of mortalities. And listen, the first time the virus struck in the year 2006. Now, since 2-3 years, what I am observing in the field, the total pathognomonic lesions we are unable to see. The significant changes we are unable to see. There are n number of cases wherein, well-managed farm, highly biosecured farm, but yet mortalities are abnormally high and no specific symptom as we used to see in the books, or as we used to see about 10 years back, 5 years back, no specific symptoms, no specific lesion, but yet on PCR and other thing laboratory tests, it is coming as H5N1. This is my reading since 2 to 3 years. Which I am seeing in most of the places, and it is a big concern. So kindly, please don't think otherwise. It's a big forum. Sorry for asking this type of a question. We have to answer ourselves. What is this no vaccination policy? Who is not doing the vaccine? And everybody knows.
Sir, I'll give the answer.
Pardon?
Should I give the answer to your question?
Yes, please. Yes, Dr. Tosh.
Since this is a zoonotic disease, first you remember what I am talking. Since this is a zoonotic disease, at the international level, the international community, scientific community thought that to control this disease, culling is the best practice, what is practiced historically also. This biosecurity measures, culling and surveillance and testing, that was practiced all over the world. It said some 15 to 16 member countries out of 182, out of one of the surveys, whatever literature available. So now what happens? So the culling policy was taken, and the no vaccination is government policy. Vaccination is going on or not, that is, I don't know. Officially, there is no record of vaccination, and the vaccination is, as I told, that this is not a simple, it is a very complex issue. To address this vaccination, as I told, there are technical issues, there are logistical issues, there are policy issues. So these issues need to be addressed. The vaccination, when there is vaccination policy developed in a country, it is not only the scientific community, it is the government, it is the stakeholders, it is the scientific community and the education. All these people has to sit together and discuss. Then a policy comes out. It is not myself sitting in Bhopal or laboratory, I will give that you go for vaccination. It is not like that. Any policy develops for control of any disease, it is the government, it is the stakeholders, it is the scientific community and the education people. They sit together and come out a policy. So that is what I want to say. And no vaccination is not my words. It is, there is no vaccination, there is policy, there is no written vaccination policy. So that's why I have written no vaccination policy in bold, because there is, whatever I tell, it is written documents only. So I think I am able to give you a reply.
Okay. Thank you.
Thank you, Dr. Tosh. Actually, one more point is that with regard to no vaccination policy, as we have discussed, I think earlier in the conference also, that the stakeholders, if you broadly classify them as researchers, industry, and the academics. So here we need to have a honest talk about the industry stakeholders, about the prevalence of the disease. The thing is, the policymakers need these data for them to be able to convince. Because any vaccination, especially with regard to diseases like avian influenza, need to have demonstrate a demonstrable economic benefit over the disease prevalence. So for that, we need a clear-cut data from the industry as well as which they have to be forthcoming with the true picture of the prevalence of the disease. Right now, sad to say that that kind of a picture is missing right now in this. So that is leading maybe to a mismatch between the policymakers and the industry, the decision making with regard to that. So that may be one of the reasons. But my question is to both the speakers is that in this situation of 2344 is actually very similar to 2006, where 2003 to 2006, where we find that the migratory birds carried the clade 2.2 virus to various countries, to around 60 countries were infected in a very short period of time. And there are huge economic losses being reported from various countries. At that time, it was maybe I think it was only vaccination was not even considered. It was considered as very few countries have opted for it. And international agencies were strictly advocating against vaccination, especially for this one. But now the similar situation is emerging with regard to 2344B virus from 2016, since its emergence in 2016. So why now the urgency for the vaccination? Is it because the spread of the virus is not limited to the Central Asian flyway, or because the other flyways are also getting into the infection? So that is the reason, because it has gone into Americas, which is a major production or exporting countries where it is more economically important. Is it now getting into that kind of a vaccination urgency is being created because of that? Could you please, both the experts, could you please explain to that?
Maybe
yeah,
Gwen, you can answer.
Okay, if I may, if I understood, so you're asking why we are globally moving towards vaccination today, right? Yeah, because
that's right.
I think people are looking at what happened the last 15 years. Some countries had a strict, depopulation policy, but where are they today? They've depopulated, and they are still in the same situation today. So depopulation cannot be applied years after years. It doesn't work. So that's one reason. Then the other reason is that with this new virus, we can't control the disease properly just with biosecurity measures. It's not anymore enough to control the spread. We need biosecurity combined with vaccination. So yeah, I think there's really a general agreement on this new requirement. And I see that in your Indian situation, it's a difficult situation because you need H5 reported outbreaks, but the problem is that people don't want to report because they would have to cull their animals, and maybe there's illegal vaccination also happening. So, I mean, it's a kind of vicious circle. How can you move forward with no incentive to report, and the need to report to be able to go ahead and to consider the disease situation? So I see a blocking situation here.
That's a nice way to interpret certain things, but actually the pandemic threat was also playing a part in decision, I think in 2006 against the thing. Now that we are reasonably confident that it's not going to evolve that much easily as a pandemic strain might have, has it led to this kind of vaccination drives because the economy supersedes the public health?
Yes, you're right. The public health threat is less less critical than in 2006, where we had many human cases, and half of the human cases were deadly cases. So yeah, I think there's far less pressure from the ministries of health, but a lot more pressure, I would say, on biodiversity, animal welfare, at least in Western countries. The general public doesn't want to see massive depopulation anymore. I mean, and what's the point of raising birds if you have to cull them? It's a big loss. So in France, that's also what people said. I mean, we're losing so many birds. We have a lot of indirect losses as well. If we start vaccinating, at least it's going to be a virtual circle. I mean, we're going to spend a lot in paying for vaccines, paying for surveillance, but labs are going to generate business, veterinarians, farmers. I mean, this is going to generate more business instead of just losses.
That's fine. Thank you.
Yeah, Dr. Nagarajan.
Yes, sir. So I think there one specific question with regard to Dr. Tosh is please give a biosecurity program to improve the vaccine performance. So I think both of you can answer. Specific. Yeah, specific biosecurity program I meant. That's what he is asking about. I think you can go through the action plan for control and containment of
action plan biosecurity.
So and then you can get more information. So I thank both the experts for their excellent information and to Dr. Tosh for a very elaborate and nice presentation. And now I invite Dr. James Kataria for his presidential remarks.
Thank you very much, Dr. Nagarajan. Really, we had a very nice presentation, though. That is the problem, which is, for many years, we have been facing, trying to control it. And what Dr. Mujib said, after 2000, still we are looking for vaccination policy. That is very true. We are looking forward. The problem is very complex. Problem is not that simple. What Dr. Tosh said, that initially what virus was isolated, and now see the change. Every time it gets changed. After 1-2 years, it is getting changed. Can you have a vaccine strain where you can do? That is a big question. Then another thing, whether that is 100% protection. Of course, no vaccine gave 100%. But even what is the range of protection we are getting with the vaccine? And then what is the excretion of virus from the vaccinated birds? Because what will happen in the farm, as Dr. Mujib said, illegal vaccination is going for 4 or 5 vaccinations. Now, okay, you can do with the birds without disclosing unofficially. But where is the cost? And how much you are spreading, do you know it? Where the virus is going from your flock? These are the questions. I mean, vaccination, jumping on the vaccination is not a solution. Really, we cannot go every time for culling. The farmer will say, okay, we cull it. What is then your solution for next time? That is true. He will ask. He needs a solution. What should I do? But you have to see the geography of the country, the poultry husbandry practices of the country. The people literacy matters a lot. If you say something, what is biosecurity? Dr. Tosh mentioned about the One Health surveillance is very important. There is an agency at the DADF, which is they are doing all these things, whether we are submitting to data. That is very important. Whether we are submitting all the disease outbreaks, important disease outbreaks, or disease happening in the farm, we are reporting to it. How the government can plan without sufficient background? Government, I think they must be waiting for it, because poultry husbandry is definitely at the stake. Every year after 2006, when I remember when the first outbreak we attended, thereafter every year it was coming, and particularly during the winter month. And as soon as the November is gone, December started, we start sweating. Now, anytime report will come out the bird. But what Dr. Tosh said, now it is around the year, it is coming up. So virus has become quite endemic. I will say it is prevalent at the pocketed. There are very pocket where it is there. Perhaps surveillance is the first key to come to some conclusion where we can go and decide for it. And mind it, again, the vaccine strain, which vaccine strain you will be using is very important. What about the cross protection when there is a variation from 2.3.1 to 2.3.4.4.B? Now you see what is the difference? Which vaccine you will work? And developing a vaccine takes time. You cannot use the vaccine strains of other country because there will be variation. And with partial immunity and immune tolerant strain may come up. What will be the answer for that? I am not saying that we should not go for vaccination. Definitely defining a vaccination policy is a must. And for that, we need to do surveillance. We have to follow the biosecurity measures in the farm. And we have a problem. You see, backyard poultry is very common in India. Everywhere you will find 5 birds, 10 birds, like that. And they don't do any vaccination. If they start dying, they cut and eat. Other birds, it will be eaten away. So that is the things which is happening. I mean, we cannot compare with the France that what they have done it, they are able to do it. Italy, they were able to do it with H7. Inactivity with small things, a small country, population of poultry is under very much control of the government agency. They can monitor it, they can do it. Sorry. So, I mean, there are many factors. Definitely anybody will get wild that they are not giving any vaccination policy. Yes, we have to give vaccination policy. And for that, we have to deal with all the agency, with the entrepreneurs, with the industry, with the government agency, with the state agency, everything, and the research scientists. What is possible? What should we do? These are the things which when we sit together and be frank, be transparent, nothing hiding, nothing, then definitely we will be having the solution. It is not like that no problem is having any solution. This problem is also have got a solution. But see, that virus is very, very notorious that way, that every time it gets changed. So we have to work continuously and report it immediately. When we have a system, we have, when 2006 only Bhopal was authorized. I am telling authorized, there was not even, they were told that you have to do it. We went with sample to the Bhopal to confirm it. And then on 18th of, I think, February, this was declared. So, I mean, and nowadays, you see, there are so many regional diagnostic laboratories, they are competent to do the initial things. That competency has to develop, laboratory competency. The field worker, they know it now, what they have to do it, they are aware, they are following it. I mean, all these things are there, but only thing is that we have to be very honest, careful for doing this thing. Definitely vaccine we require for the control, but we have to do the ground. I think this is what I want to say to Dr. Mujib and Dr. Govind and Dr. Tosh. Of course, he has spoken it very nicely. Thank you very much.
Thank you, sir, for your nice remarks and summarizing the context of this particular web series. And may I have the pleasure of inviting Dr. Chatterjee, Director, ICAR-DPR, Hyderabad, for his remarks. Sir, stage is yours.
Thank you. Thank you so much, Dr. Nagarajan. I think this was the great issue as regards to the poultry sector of this country is concerned. And it remained the main issue for the last almost 17 years, since 2006. And my esteemed colleague, Dr. J.M. Kataria, he was associated since 2006, I think, with the whether we should go for vaccination, and then how to, control the disease when it was there in 2006 and thereafter also. That time he was in the DASD or DADF, Ministry of Agriculture, and he contributed a lot. And not only that, there is a committee of Ministry of DASD, whether you should go for vaccination for any disease or not. For this particular disease, Dr. J.M. Kataria was there sometime, and Dr. M.R. Reddy, my colleague from my institute, he was also there. And there was a confusion earlier whether you should go for vaccination or not. But I will not go in that way. But recently, LPAI vaccination that has already been, it is going on, and that has been recognized by Government of India also. So some relief is there. But the thing is that why we are unable to develop the vaccine for HPAI. I think in many countries, many laboratories, they are trying to develop the HPAI vaccine through new technologies, as our speaker from France, she said very clearly globally what is happening as regards to HPAI and LPI, and what vaccines are being used, and how China, they have contained this particular disease through vaccination and all that. But I think I have heard 2-3 months back, there was one meeting in Paris itself of World Animal Health Organization. And there it was discussed about whether we should go for the development of vaccine for the HPAI, and that was, and a green signal has been given. And the new technologies like genome editing and all that, they are being used probably. And NISAID also is also trying, as I have heard, for development of vaccine of HPAI. But again, there are certain issues whether you should go for HPAI vaccine, because HPAI is related to One Health. It is related to, it will infect the human being. And whether the mutation, as has been mentioned by Dr. Kataria very clearly, that it is a very notorious disease. And when the mutation will happen, in which strain, we do not know. So surveillance is the most important part. Although unofficially the poultry sector or the poultry farmers, they are doing the LPAI vaccination here and there. But we do not know. We do not know which particular strain is there and which particular strain we should use for the vaccine. So for that purpose, surveillance is very much required. And in that surveillance, which particular strain combination is there in that particular area, that is also required. Then we can go for the vaccination for that particular strain accordingly in that particular location. However, there is a committee of compartmentalization by, again, DAHD. Earlier it was with Ministry of Agriculture. Now this compartmentalization committee is the Ministry of FASD, where I am a part for the last 13 years. Earlier I was a member, now I'm a chairman of some committee of compartmentalization, and the export that was hampered earlier when, this avian flu has occurred 2006 and thereafter. But now there is a process and that is being certified by the Government of India, Ministry of FASD, and there is a specific committee. They are taking care of all the aspects. And not only that, the people or the country who are taking our products of the chicken or the egg from our country, they are importing, they are also coming, they are also seeing if there is any loophole or if there is any problem, then they will finger on us. But one thing has happened recently, that Sri Lanka and Malaysia, they have also come forward and they are also importing our chicken or egg products from our country. Besides, Gulf countries and all that, and egg powder is going to Europe and Russia. But I always say in different forum, our colleagues from the poultry sector, that you please give emphasis on the export of the poultry product. We are already only having 800 crores export value from the poultry sector every year, which is to be increased further, because domestic market, there are many problems. We don't get the market for eggs as well as for chicken. Many times, many farmers, they are at loss. So we should find out, or we should explore the export market. And there is a procedure, there is a framework by Government of India, and the state government also should come forward. Because I know the Tamil Nadu government as well as the Maharashtra government, they are they're taking all the initiative, and they are very cooperative in that particular aspect to the private sector. And after initial screening by the state government, both Maharashtra and Tamil Nadu, that is going to the DHD, Ministry of FASD. But I do not know why Telangana and AP, they are lagging behind in that particular aspect. So that is, of course, not a part of avian influenza aspect. But I am very much optimistic, and I am very much hopeful. We have contained corona all through the world, but why we cannot contain the H5N1 or the other HPAI strain combination? So probably maybe another 2-3 years, we will come out with some vaccine through which we can contain both the LPI as well as HPI. But for that, already Dr. Dey has mentioned that two things are required. Only vaccine will not protect. It is the combination of vaccination as well as the biosecurity measures that has to be taken care of. So my sincere thanks and compliments to both the speakers and AAHP as well, the President, Dr. J.M. Kataria of AAHP, and Dr. Reddy, my colleague from this particular institute. Thank you so much.
Thank you, sir. Thank you so much for your nice insights into the policy decision-making process. May I invite Dr. D.K. Dey, Vice President of AAHP, for his remarks.
Good afternoon, everybody. So I have nothing specific to say. I think the only signal I'm getting that time to work more coherently, industry, academia, policymakers, to find a sustainable solution. Having said that, I know for sure solution is not overnight. We have to have a strategic approach towards it. We have expertise, global expertise, local expertise, and keeping our national interests in prime, we can exchange knowledge, because most of us have a lot of visits overseas, what is happening in a neighboring country, the positive side, negative side we can share. So I think my request to AAHP that debate on the issue, let it not be limited to one webinar. We can continue to discuss on it and supplement government for the best interest of the nation. That's what from my side. Thank you so much.
For your remarks. May I invite Dr. Sanjay Gaukare for his remark, and Vice President of AAHP for his remarks, please.
Yeah, good afternoon, everyone. Thanks, Dr. Dauphin and Dr. Tosh for excellent presentation. And you have covered large HPAI related issues, including the virus structure and changing the virus nature as well as the control strategy. When we talk about the HPAI, there are two clear fear factors we need to address. One is the fear in the scientific community. So it has been since so many decades we are hearing that it is a communicable disease to the human. And with only that fear factor, the dialogue which has to be taken in front of the policymaker and the industry will not happening, and that only the fear factor has a big hindrance to evolve a policy. Number two, second fear factor is in the minds of the farmer. When there is a HPI, there is a culling policy, start culling, then there is economic loss, prices go down, the farmers is at risk. So these two fear factors needs to be addressed when to evolve a policy. Now it is very much visible and clear that the depopulation is not the clear answer to the HPI. One has to have vaccination policy. Like so many years we have now, after so many years we have established now for the vaccination policies for the LPI. Lot many papad has to be built for this LPI policy. Similarly, we can have cohesive approach with the government, the industry stakeholders, as the academy, the Bhopal, the excellent lab in the world, I can say, because I know you people, you are doing excellent work, and with that, definitely we can evolve some policy, vaccination policy, as well as the surveillance policy against the HPA also. India is changing, that there is a huge change in the mindset of the policymakers also. So the industry and the policymakers are coming together. It is a positive sign. And with the help of the government agencies like Bhopal Lab and the regional state laboratories now well equipped, we can evolve a good surveillance structure also, as well as the vaccine monitoring policy, if at all this vaccination policy get to be evolved. So there is always support from the industry, so that these two fear factors, if at all it's to be removed, then industry also come forward and we'll have a cohesive approach to contain the HPAI. Thank you very much.
Thank you, Dr. Gawkari, for your nice remarks. Basically, it is a situation of whether egg comes first or the chicken comes first. The policymakers need the data, whereas the industry fears that the real data may lead to huge economic losses for them. And also the fear in the minds of farmers about the compensation, whether the government will be able to deliver it in the proper way or not, and whether they will be adequately compensated. So those issues need to be addressed. But of course, as you said, for a dialogue to happen, all the three arms of decision making should sit together and evaluate and make a roadmap to how to go forward, so that we can be a win-win situation for all the stakeholders. So thank you all.
Dr. Nagarajan,
Sir?
Dr. Nagarajan, just one thing.
Yes, sir.
There is a committee, all of you know, in FASD ministry. They are giving compensation for the birds when there is HPAI, the birds which we are culling. So it is being changed, the value of the compensation for each bird almost every year by the FASD ministry. And recently, there is one committee on that. And I am also there in that committee. And the private partners, they are also there in that committee. So recently, we sat together in Delhi, and we have increased the compensation amount for the cull birds in case if there is HPAI anywhere in the country. So there is a huge increase in that compensation amount. So I think that will come maybe in the near future. What is the compensation amount for the birds to be culled? So probably that will solve to some extent for the farmers. Thank you so much.
Thank you, sir. Thanks for your nice information.
Thank you, Dr. Chatterjee. What compensation, but you know, that is for commercial is okay. What about your birds, which have gone for years?
Yes, sir. Yes, sir. You are very right. You are very right.
Compensation can compensate your efforts.
Yeah.
So that is the situation, Dr. Gawkari.
Exactly.
Fear among the scientists is this one only. Because once that virus go in silent mode, that will be disastrous. So we have to be very careful about the surveillance, active surveillance of all the things, bird, pond, poultry farm, government farm, everything. I remember Chandigarh, when outbreak was going on, Chandigarh farm, they are having many birds. They were so much scared that what will happen to our birds. We said, just close your facility for the visitor. Nothing can be done. Just lose the visitor, no problem. But you cannot go for vaccinations, because that time India started vaccination with the Netherlands having a vaccine buffer. That they will keep the buffer yearly. We will pay 30 crore rupees to them for keeping the vaccine doses. But now it has been discontinued and it was never been used. So these things, there are many factors perhaps. Fear is due to virus and many more things. It is not in the mind of the scientists or the industry or anybody else.
Sir, you have pointed out very nicely what about the pure lines and the parents.
Yeah.
What will happen with that? What we have done in our institute, all the pure lines, whatever we have developed through breeding and selection, we have duplicated those birds. But the thing is that if the same HPAI that happens together at the same time in 2 different locations, why do we have kept the bird? So that will be a problem. But we have duplicated, and for industry also probably they have duplicated their parent lines. And parents, of course, there is a problem. And for parents also, we have taken care of about the compensation amount. It is higher, very higher. But that cannot, the amount that the farmers, they will lose. That is not to that magnitude.
Thank you, sir, for an excellent interactions and discussions. Thank you all. Thank you to both the speakers. But for the formal vote of thanks, I invite Dr. T.R. Kannagi.
Good afternoon, all. Yes, sir. Good afternoon all. On behalf of AAHP, I thank both the speakers of today's webinar, Dr. Gwenaëlle and Dr. Tosh, for their wonderful comprehensive presentation. I also profoundly thank our President, Dr. Chakradhar ji, for motivating us continuously, and also the Vice Presidents and other EC members. Hope all the participants enjoyed and benefited by today's webinar. I also thank our beloved Director, Dr. R. N. Chatterjee ji, for giving latest insights on this topic. Thanks to the DPR team for helping in live streaming the webinar online through social media platform. Thank you so much, one and all. You have a great day. Thank you so much.
Thank you, one and all. We'll be back with the next series. So everyone is invited for that, and we will be informing as and when it is finalized. Thank you. Thank you so much.
One point I would like to mention other than this webinar.
Yes, sir.
Yeah, next we will be having another webinar, maybe 1-2 months. But as you all know, all the member of AAHP and industry, that we are going to have our conference in the month of February in Hyderabad. So I request you all that you encourage your colleagues, scientists to participate and join that conference. I also request Govind to, if she can make it, it will be very nice. Of course, from our country, there will be many. And thank you very much. Please do participate in that conference.
Thank you.
Plan it now. Thank you.
Thank you, sir. Thank you. And this has actually provided the basic platform for integrating the academic and the industry. It's a very good platform. Probably in the next thing, we can also try to include a policymaker also, stakeholders also into our webinar series, so that they are also better informed of this. So thank you, sir. Thank you so much for the AHP and also the experts. Thank you so much. Nice meeting you. And thank you so much. Bye.